In this article, the researchers studied on the amount of the ascorbic acid content in various pharmaceutical samples using bromocresol purple. This study introduced a new approach of spectrofuorimetric method to study the interaction between bromocresol purple and ascorbic acid for the first time, as no findings were reported in the previous studies. The experiment involved optimizing various experimental conditions for the proposed method. After validating the proposed method, the study determines the amount of ascorbic acid in 15 pharmaceutical samples, including tablets and powders. The study also investigates the potential interference from other substances that affect the fluorescence signals of bromocresol purple that could impact the accuracy of ascorbic acid measurements in the samples. By analyzing the analyte, which is the ascorbic acid in the sample, is crucial for accurately determining its content. This is essential to ensure the quality, safety, and effectiveness of the pharmaceutical products. The proposed method provides a practical approach for achieving this goal. By identifying variations in ascorbic acid content helps to identify issues like degradation and interaction among active ingredients. It also aids in monitoring storage conditions, shelf life, and expiration dates. As a vital nutrient for humans and some animals, ascorbic acid could serve various biological roles such as it serves as antioxidant, enzyme cofactor, and gene expression modulator. Insufficient dietary intakes of this vitamin C could result in various health issues. The study aimed to create a straightforward, reliable, quick, sensitive, and cost-effective method for this analysis. The researchers chose this spectrofluorimetric method over spectrophotometric method due to its simplicity, high sensitivity, and applicability to a wide range of samples. In addition, this method is more preferable than UV techniques because it is more sensitive, less influenced by interference, and requires less sample preparation. Although ascorbic acid does not fluoresce, bromocresol purple was used as a chromogenic reagent. This facilitated the measurement of ascorbic acid content by generating a colored compound, which is detectable using a luminescence spectrometer.
Advantage of the analyzed spectrophotometry offer a significant advantage over spectrophotometry due to high sensitivity. Namely, the analysis of the nanogram level of the target analyte expect a sample containing a different compound, which need removal before analyze the sample. Preparation is straightforward. This method relies on the concept that the analyte being studied can inhibit fluorescence or undergo a reaction with another substance to generate a fluorescent product. This alteration in fluorescent intensity observed upon the addition of the analyte manifests as increased decrease or marked enhancement in the fluorescent signal intensity. For the further research, could involving implementing this approach as an actual sample or experimenting with the introduction of certain component or metal ion that will impact the reaction, aiming that it has reproducibility. Reaction rate expand the linearity range and potentially streamline sample preparation. A limitation of the method is the narrow linearity range. Table 7 provides a comparison of performance characteristics including linearity range and limit of detection. Among various spectrophotometric methods, the analysis revealed that some methods inhibit a narrow linear range, while others have a wider range. In terms of linearity, the proposed method, when compared to methods in the literature, threshold as it does not offer a wide field of linearity as indicated in Table 2. Despite this limitation, the detection limit presented in Table 7 is not superior to several other proposed methods for determining ascorbic acid in pharmaceutical sample. This method shows promise for the straightforward and relatively swift determination of ascorbic acid, making it applicable to a variety of pharmaceutical samples containing diverse additional components. Information provided from the spectra. The study involving screening, excitation, and emission spectra of the reagent phosphorus buffer (PCP), both with and without the analyte ascorbic acid. Initial scan without ascorbic acid show excitation peak at 300, 381, and 509 nanometer, and emission peak at 320, 361, and 641 nanometer. When ascorbic acid was introduced at the concentration of 4.65 times 10 power of minus 4 mol per liter. A noticeable decrease in fluorescent intensity value was observed across all beads. The most significant reduction occurred at 318 nanometer excitation wavelength from 895.83 to 578.75 and 641 nanometer emission wavelength from 1000.07 to 826.60. These specific wavelengths were then selected as the working wavelength for the method.
The information obtained helps to identify optimal parameters for the proposed method to analyze the ascorbic acid content in 15 pharmaceutical samples quantitatively. The spectra produced from the optimization step helps to determine the working excitation and emission wavelength for the spectrofuorimetric quantification of ascorbic acid, which is important for measuring the fluorescent intensity accurately. The calibration curve established during the optimization of experimental conditions shows a linear relationship between fluorescence intensity and the concentration of ascorbic acid. The data from the optimization step was applied in the proposed method for quantifying the analyte in the sample. This involved comparing the amount of ascorbic acid found using the developed method with the label value. The changes in the fluorescent signal intensity were also examined for the presence of interference substance in the samples.
Relation of the analytical procedure. The precision of the spectrofuorimetry method was improved by analyzing ascorbic acid solution within a concentration range of 0.0480 millimole per liter to 0.0058 millimole per liter. To assess precision and repeatability, the fluorescent intensity of the prepared ascorbic acid solution at 0.477 millimole per liter was predictably measured in 4 replicates with n equal to 10. The obtained result when just to calculate the standard deviation SD and percent RSD revealed a measurement precision as well as RSD of 8.46% for this concentration. Overall, the method inhibits commutable precision and repeatability, along with satisfactory recovery value indicative of accuracy.